This abstract will summarize the paper Anti-diabetic drug binding site in KATP channels revealed by cryo-electron microscopy. KATP channels are octameric protein complexes consisting of 4 KIR6 and 4 SUR or sulfonylurea receptor subunits, which respond to the binding of ATP. In pancreatic beta cells, KATP channels consist of KIR6.2 and SUR1 subunits. Within these cells, KATP channels close in response to a rise in intracellular ATP concentration resulting from glucose metabolism. Closure of the channels causes depolarization of the cells, which in turn leads to calcium influx and ultimately the secretion of insulin granules to the bloodstream via exocytosis. This is an important mechanism for the control of blood glucose levels in humans, and mutation of these channels can lead to a variety of issues such as type 1 diabetes, wherein a person's ability to regulate their blood glucose level via the secretion of insulin is significantly diminished. Sulfonylureas such as glibenclamide or GBC are drugs which are known to block the efflux of potassium ions from KATP channels in pancreatic beta cells, thereby stimulating the secretion of insulin by the aforementioned mechanism. It is therefore used in the treatment of type 2 diabetes in order to aid in the control of high blood glucose levels, a symptom which is ordinarily observed in patients suffering from this disease. Despite its importance as a therapeutic agent, the mechanism by which GBC inhibits the action of KATP channels remains unknown. In order to elucidate the mechanism of interaction between GBC and KATP channels, the researchers in this study expressed the SUR1 gene from a hamster and the KIR6.2 gene from a rat in a rat cell line, and the channel proteins were purified. Single particle cryogenic electron microscopy was then performed, and a model was constructed with a resolution of 3.8 angstroms. Finally, site-directed mutagenesis was performed to convert likely GBC binding residues to alanine in order to elucidate the effect this had on the sensitivity of these proteins to GBC inhibition. The model confirmed previous findings that the ATP binding site was located at the interface of the N-terminal domain and C-terminal domain of adjacent KIR6.2 subunits, appearing in green in Figure 1. It was postulated based on this location that the mechanism by which ATP prevents the efflux of potassium ions through the channel involves prevention of movement of the KIR6.2 N-terminus relative to the SUR1 subunit, which is believed to be necessary for potassium ions to pass through the channel. Despite previously held notions, arginine residues 201 and 301 do not interact directly with ATP, but play a structural role in stabilizing the binding residues, as shown by its proximity to these side chains in Figure 2. It was previously believed that the GBC binding site was located near tyrosine 230 and tryptophan 232 due to a lack of high-resolution models. Due to the high resolution of this model, researchers were able to observe a region of non-protein density that was believed to be GBC. This demonstrates that the GBC binding site is located within the transmembrane bundle of SUR1, as shown in Figure 3, with 6 residues from transmembrane domains 1 and 2 contributing to binding. It appears that tyrosine 230 and tryptophan 232 instead play a role in stabilizing the binding residues, which is demonstrated in Figure 4, as they are located too far away from the GBC binding site to participate directly in binding. This explains the apparent importance of these residues in previously performed functional assays. Finally, site-directed mutagenesis was performed on the 6 postulated binding residues, and KATP channel activity was measured using a rubidium efflux assay. The percentage efflux of potassium is shown in Figure 5 for each mutant in various colors and compared to wild type in black. It was determined that mutations at all 6 binding residues reduce the sensitivity of the channels to GBC inhibition at 100 nanomolar GBC concentration, with 4 of these mutants displaying significantly decreased inhibition up to 1 micromolar concentration. This confirms the importance of these residues in GBC binding. In its entirety, this study provided clarity to researchers in the field as to the mechanisms of both ATP and GBC binding to KATP channels, and provided strong evidence of this through functional assays. Although only GBC was employed in this study, these findings may relate more broadly to the mechanisms of action of other sulfonylureas. This will assist researchers in the field in developing drugs designed to target other subtypes of potassium channels in the treatment of various diseases.